morning, and I also had the opportunity to serve as the translational co-chair with Joe Califano, and I wanted to thank the leadership for that opportunity. This morning, I have the pleasure of introducing Dr. Mack as our keynote lecturer. Dr. Mack is currently the director of the Campbell Family Institute of Breast Cancer Research at Princess Margaret Hospital and a professor at the University of Toronto in the departments of both medical biophysics and immunology. Prior to his present position, he received his master's in biophysics from the University of Wisconsin and his PhD in biochemistry from the University of Alberta. Dr. Mack's work has been widely recognized, both in Canada and abroad, where his scientific achievements have had a major impact on public health around the world. His legacy includes a series of critical discoveries in fields as diverse as immunology, signal transduction, and cancer biology. Dr. Mack became an internationally acclaimed immunologist in 1984 with his discovery and cloning of the human T-cell receptor, solving one of the toughest problems in immunology. Until then, the nature of its antigen receptor had been unknown. Dr. Mack discovered that T-cells actually use an entirely different set of genes for antigen recognition. He showed that the T-cell receptor for antigen was in many ways analogous in design to antibodies, yet totally different in genetic origin and sequence. The T-cell receptors are possibly the most important element of the immune system. They allow T-cells to distinguish self from non-self. Identifying this gene may lead to better treatments for a host of immune disorders and other related conditions, from diabetes to cancer. It is impossible to understate the importance and impact of this seminal discovery on all fields of immunology. As director of the Campbell Family for Breast Cancer Research Institute since 2004, Dr. Mack international recognition by identifying critical molecular pathways that are expressed in cancer cells, which begins to explain how these cells escape normal behavior in terms of the control of growth and death. He has established one of the finest academic drug discovery engines, having identified 2 novel drug targets, particularly relevant in today's times. Dr. Mack holds honorary doctoral degrees from numerous universities in North America, Asia, and Europe. He is one of the most honored scientists in Canada and is an officer of the Order of Canada, a member of London's prestigious Royal Society, a recipient of Canada's Gardner International Award for Science, a fellow of the Royal Society of Canada, and a foreign associate of the National Academy of the Sciences from the USA. He has also received many highly distinguished prizes, including the Novartis Prize in Immunology, the Paul Ehrlich Prize, and the Sloan Award. Dr. Mack is the author of countless high-impact scientific manuscripts and review articles. Most recently, in work just published in the prestigious journal Nature, he led a team of international scientists that identified a causative link between the product of a mutated metabolic enzyme and the onset of acute myeloid leukemia, one of the most common types of adult leukemias. Called an oncometabolite for its role in cancer metabolism, this is the first example of how a metabolite can cause cancer and sets the stage for future work to develop inhibitors as a novel form of cancer treatment. Again, a novel insight and major contribution. Today, Dr. Mack's talk is titled Cancer Metabolism, Back to the Future. Please join me in giving him a warm welcome.
Thank you, Dr. Rocco, for this kind introduction. I also like to thank the organizers, especially Drs. Irish and for this kind invitation. So what I like to do today is to give you somewhat of a conceptual viewpoint of personal conception viewpoint as to where we think we're going in the treatment of cancer. So not so far long ago, about 350 years ago, a French playwright pretty much described the somewhat, I would say, I wouldn't use the word barbaric, but somewhat crude way by which we are treating our patients. And he said, doctors pour drugs of which they know little to cure disease of which they know less into patients of whom they know nothing. Now, it's now 350 years, and somewhat we're still struggling with some of the comments that he made. Nonetheless, I think the necessity of these treatments somewhat justify the kinds of usage. For example, in 1942, Louis Goodman and Al Gilman did nothing less than treating lymphoma patients with nitrogen mustard, which is, of course, a chemical warfare agent. Now, this had basically done a lot for different kinds of cancers, and chemotherapeutic agents basically rule from the point of view of drugs during the 50s, 60s, 70s, 80s, 90s, and up to today. However, you can see from the list of agents that have been used that by the turn of the century that we realized that we have actually hit the wall, and very few, if any, pharmaceutical companies today are actually developing chemotherapeutic agents. The idea, of course, was that if we were to be able to understand the origin of cancer, then it would be possible for us to get more targeted treatment and to prevent the collateral damage that accompanies chemotherapeutic agents. But this actually did not happen as early as we thought. Even back in 1966, the Nobel laureates gathered in Lake Constance, Germany, and basically asked themselves, what are we doing to find out the origin of cancer? And at the end of 3 days of deliberations, talks, the Nobel laureate Otto Warburg actually made the conclusion that cancer by 1966 was not caused by viruses, was not caused by genes, but it was caused by the replacement of the respiration of oxygen in normal body cells by fermentation of sugar. Now today we know that cancer can be caused by viruses, and we heard that so elegantly from Zurhausen yesterday. And we know that cancer is caused by mutations of genes. But cancer is also caused by the replacement of respiration of oxygen in normal body cells by the fermentation of sugar. And that is where I would like to take the rest of the talk. In 1976, the discovery of oncogenes by Stalin, Varmus, Bishop, and Vogt basically set the stage that as molecular oncologists would like to develop drugs, the only path was to target oncogenes, because if oncogenes cause cancer, then it is logical for us to target oncogenes as it would then cure cancer. And the idea up to today is to shoot the horses, because the horses are the oncogenes and the tumor suppressor genes that basically drive the consequence of cancer, and that we thought was actually quite accurate for a little while. You can see that targeted therapeutics came into play approximately a dozen years ago with the discovery that HER2 antibody is able to actually prolong the survival of breast cancer patients with HER2 amplifications, and also that Gleevec were able to prolong the survival of chronic myelogenous leukemia patients, and EGFR inhibitors came in. But by the year 2007, it was obvious to the industry and to many of us that we have somewhat hit the wall again. Between the year 2007 and 2011, not a single major cancer target drug was actually approved by FDA. And the interim, 2 immunotherapeutic agents actually made it to the list. Today, we still are encountering a tremendous amount of enthusiasm, but in my words, in my thinking, I think it's time for us to also be a little bit more cautious. For example, take the case of squamous cell carcinoma of the head and neck. You can see here, after sequencing thousands of patients, the discovery was that there are very few recurring oncogenes. This is not actually unique to head and neck cancer, but many other cancers, like ovarian cancers, neuroblastomas, pancreatic cancers, with the exception of RAS, there are actually very few recurring oncogenes. Moreover, the oncogenes like mutated RAS had been basically the graveyard for many different pharmaceutical companies who seek to inhibit this particular function. The lack of recurring oncogenes is a problem not only for head and neck cancer, but it is a problem for many, many different cancers. And I think this is the problem we have today. Nonetheless, that doesn't mean that we do not have limited success in making differences in the clinic. For example, the targeting of head and neck cancer with the inhibitors to the EGFR as a therapy has actually made some headways. As you can see here, that in this landmark trial of the Bonner trial, that radiation plus or minus inhibitor to EGFR in locally advanced head and neck cancer, the overall survival is actually significant. I would not say it is a grand slam home run, but of course there are very few grand slam home runs. Now, the elephant in the room, of course, as we now know today, that there are 300,000 mutations known. 20 of the 23,000 genes are now known to be mutated in the epithelial cells. There are 50 to 100 drivers, and leukemia lymphomas 20. There are thousands of amplifications of genomes. In breast cancer, it is estimated that 70% of the whole breast cancer genome can be amplified or deleted. There are translocations, there are epigenetic changes, there are hundreds and hundreds of drivers. 90% of these drivers are loss of functions, and therefore it is very difficult to target a gene that has already lost the functions. To make it worse, we have been told recently by Charles Swanton that at least in kidney cancer, that both intra and intertumoral heterogeneity occurs at a significant level, and making it difficult for us to target these cancers. One could argue, however, that there are only 12 different pathways that govern these deregulations due to mutations or viral infections, and that we could figure it out by a combination of drugs. And today, the paradigm is personalized medicine, where we will sequence the whole patient's genome, we will know all the defects, we will make drug combinations based on pathway knowledge, or sometimes intuitive deduction. But in many cases, I have to admit that we are simply just tossing the coin, as these 12 different pathways that are affected are totally intertwined, and we have no idea whether to target upstream or downstream. So then comes, I think, the future. In my mind, the future, we have to target the cart. In other words, we cannot afford to shoot all the horses, since 90% of the horses we cannot even see, and the rest of the horses are spread over so many different cancers. And many cancers have no drivers that are oncogenes, mostly tumor suppressor genes. What about then thinking the concept that we will target cancer by targeting the consequences? Consequences are the phenotype and the changes of the cancer irrespective of whether it is viral infection, or whether it is epigenetic changes, or whether it is oncogenes, or different tumor suppressor genes. The immune system, for example, has been quite helpful in this regard. For example, anti-CD20 rituximab have been treating non-Hodgkin lymphomas for many decades now, and more recently, the activation of T lymphocytes, the CD8 lymphocytes, to attack tumors are actually coming to fruition. Scientists have known for decades that immune cells infiltrate the tumors, and it's always thought to be there for no reason. However, there's been a whole series of trials that suggest that immune cell infiltrates most of the time predict good prognosis in breast, liver, colon, ovarian, melanoma, non-small cell lung cancer, hepatocarcinoma. So if the immune cells can actually kill the tumor cells, why do they stop? Well, because maybe since the immune system is actually trained to only attack non-self, that most of the tumor cells are actually self, and so that may be a problem. Now, this is actually buttressed by the finding that there is improved survival of patients with papillomavirus positive head and neck squamous cell carcinoma. In a prospective clinical trial, it's shown that patients who are actually HPV positive actually do better, suggesting maybe the immune system is recognizing components of the virus. Now, the immune system, just like a car, has accelerator and it has brakes, and these brakes are called negative feedbacks or checkpoints. And there are genes like CTLA4 and PD1, which are the brakes or the negative checkpoint. Our laboratory discovered the CTLA4 function in a paper in 1995 in Science, that the mouse without CTLA4 dies within 2 weeks of lymphoproliferation. The mouse, at the end of 2 weeks, 20% of the total weight of the mouse are T lymphocytes that lead to the demise of the mouse. Jim Ellison and others took this concept and developed an antibody against CTLA4, thereby removing the brakes, allowing the T cells to continue to rage and rage and rage. This, of course, had led to the treatment of stage 4 melanoma patients, metastatic, with a very significant increase in survival. But at preliminary data, as you can see from the tail of the Kaplan-Meier curve, it suggests that maybe even some patients have very, very long survival because of this treatment. Now, this treatment completely disregards the oncogenes and the tumor suppressor genes, as they only care about the antigens on the tumors. Exhaustion. T cells, like all of us, get exhausted, and what can we do to add to this particular battle against tumors, that the T lymphocytes will continue to rage and rage and rage? In collaboration with Pam Ohashi in our institute, we discovered that a growth factor called interleukin-7 can actually prolong the life of a pancreatic cancer in mouse for approximately 100 days. This treatment of interleukin-7 has now entered the clinic in phase 1 clinical trials. Let me just briefly touch upon another, which I think would be the cart of the future, and that is aneuploidy. Aneuploidy has been known for over 100 years as a hallmark of cancer. That cancer, by and large, cells have 70 to 90% of the cancer cells have somewhere over 46 chromosomes, all the way up to over 100 chromosomes. This is, of course, a difficult task for cancer cells to carry out as well, as aneuploidy in normal settings can only be tolerated in the case of chromosome 21, and of course, that is Down syndrome. Cancer cells, in this particular case, I can show, it's actually showing approximately 70 chromosomes, and yet they're able to struggle through end up dividing its 70 chromosomes at the same time. I said the word struggling because they do make a lot of mistakes. And trying to understand the clue of these mistakes, our laboratory, together with the Campbell Family Drug Development Institute, have developed 2 molecules, one hopefully to enter the clinic next year, against aneuploid cells. As you can see that on the graph, there is compound 1 and compound 2. Compound 1 targets the target TTK, and compound 2 targets the target PLK4. In this case, the compounds were able to prevent the successful mitosis, and in the case of compound 1, got the segregation of the chromosomes. In the case of compound 2, we were able to prevent the centrosomes from aggregating into 2. Since it cannot aggregate into 2, the cancer cell dies. In my last few minutes, 10 minutes or so, I'd like to turn to the field of metabolism. Back in several hundred years ago, Pasteur had showed that the organisms in the absence of oxygen actually will do glycolysis with the production of lactate. In the presence of oxygen, they do oxidative phosphorylation. Now, Warburg, about approximately 100 years ago, discovered that cancer cells, even though in the presence of oxygen, they actually do glycolysis most of the time, which is a paradox in the sense that if cancer cells actually really needs more energy, why would it forego 34 ATPs and only stay with the 2 ATPs that you net gain from glycolysis? Today, we understand that the Warburg effect is actually driven none other than the oncogenes and the tumor suppressor genes. Oncogenes like Mack is actually able to drive the influx of glutamine into a cell, and then subsequently RAS, PI3 kinase, HIF1 alpha, other oncogenes actually drive glucose into the tumor cells, and of course that sets the stage for the PET scan. Cancer cells adapt to different metabolism driven by oncogenes and tumor suppressor genes for 3 reasons. 1, they want more ATPs. Second, they want more building blocks. But very importantly, they want more redox because of the amount of reactive oxygen generated in the cancer cells by new metabolisms by oncogenes, and as well as by the increase in protein translation. Here is an experiment that we did together with the company Avail to ask the questions as to since the EGFR receptor-driven and also other receptor tyrosine kinase drive the activation of both the PI3 kinase pathway and the RAS pathway. The question is the driving of this pathway addicted to metabolism because the PI3 kinase pathway is responsible for glucose activation, glycolysis, etc., or are they actually not important at all? So in order to test this, we test 211 breast cancer samples and treated them with rapamycin, which is the homolog of which, of course, is CCI779 and RAD001. And we performed this in vivo resistance screen. In essence, you put 211 breast cancer samples into mice, and then you treat them with rapamycin. As you can see, this waterfall plot very nicely shows a continuum of the effect of rapamycin on the growth or the inhibition of growth by rapamycin. See that the mTOR response are the tumors that have shrunk in the order of 70% or more at one end, and the other end, there is no change. So in order to find out what each one of these tumors actually harbor genes that confer resistance or sensitivity, we performed a microarray of 40,000 probes on these 211 tumors. The outcome was actually quite stunning. The only genes out of 40,000 probes that we got from this particular screen of 211 tumors was a gene called carnitine palmitoyltransferase 1C. As you can see, that the corresponding to the response, many of these actually did not respond, but those that responded, none of them actually expressed this carnitine palmitoyltransferase. Carnitine palmitoyltransferase is, of course, a gene that is able to take acyl-CoA fatty acids into the mitochondria for beta oxidation. As you may imagine, this is what cancer cells can do. They go for ATPs, building blocks, and redox. Just sometimes they would actually take lipids for these purposes. But the surprise is that it was the 1C form instead of the 1A and 1B form the cancer cells have reached out to help the cancer, the adaptation. And the 1C form is particularly surprising because it is a brain-specific form of the carnitine palmitoyltransferase and is found in the peroxisome. Only 10 papers have ever been published on this gene as opposed to thousands published on 1A, found in most tissues, 1B, found in the heart and the muscles. In fact, even though they are specific for the brain in normal tissues, cancer cells going from neuroblastomas to sarcomas to leukemias, lymphomas, head and neck cancers, pancreatic cancers, liver cancers, all upregulate this gene so they can actually get hold of more ATP, more building block. But in our mind, one of the important parts it is going after is that this is a redox gene, which is capable of taking care of reactive oxygen in addition to providing ATP and building blocks for the cancer cells. Then brings us to the model that metabolism oncogenes activate reactive oxygen, which at two very high level, the oxygens, reactive oxygen would kill the cells or put the cells into senescence. So what cancer cells do is they upregulate a whole series of redox genes in order for them to survive. And as they survive, they adapt to these genes and they become mutagenic and how cancer actually comes forth. That was carnitine palmitoyltransferase 1C. Another example, a couple of slides to demonstrate that, is the discovery by our laboratory that the Parkinson's disease gene number 7, which is a DJ1, found in a Drosophila fly screen against cell death, done by Malte Peters, who is now the head of clinical oncology in Novartis in Basel. This gene is a 25 kilodalton molecular weight gene. This gene is a Parkinson's disease gene. Parkinson's disease gene, 100% penetrance when patients have loose DJ1 or PARC7. The underlying mechanism is that of redox again, that mice without DJ1 or PARC7 treated with an oxidative stress agent have their dopaminergic neurons disappear at much faster rate, leading perhaps to the cause of Parkinson. However, the overexpression of PARC7 correlates with bad prognosis in cancer, lung cancers, ovarian cancers, gynecological cancers, esophageal cancers, et cetera, et cetera, et cetera. What does DJ1 do? DJ1 controls the master transcription regulator of redox, a transcription factor called NRF2, which is able to activate a lot of genes that can sequester the reactive oxygen. And knocking this down would kill the cells. In my final 3 minutes, what I will tell you is NADPH. NADPH is a molecule that can convert glutathione to glutathione disulfide into glutathione. There are only 3 pathways to make NADPH in man, mouse, yeast, or crocodiles. And they are the pentose phosphate shunt and glutaminolysis. And the final pathway is when the enzyme isocitrate dehydrogenase is converting isocitrate to alpha-ketoglutarate. In a very, very surprised and stunning paper by the Vogelstein's group in 2008, the field discovered that mutations of isocitrate dehydrogenase, arginine 132 histidine, is found in 12% of the glioblastomas. In fact, this mutation is found in 80 to 90% of gliomas and secondary glioblastomas. It is found in AML, 20%, angioimmunoblastic T-cell lymphomas, non-small cell lung cancers, et cetera, et cetera. What does this particular mutation do? Our laboratory and that of the laboratories of Craig Thompson and also Agios Pharmaceuticals discovered that the mutation of isocitrate dehydrogenase drive alpha-ketoglutarate into 2-hydroxyglutarate. You can see here from glioblastoma patients, secondary glioblastoma, they have in the order of 100 to 1,000 times more of this particular metabolite in their tumor cells. In order to understand this, we make a point mutation, knocked it into the mouse, and you can see that brain abnormalities is beginning to appear. But significantly, we were able to, in the myeloid series, create a mouse with similarities to myelodysplastic syndrome, and that with a very dramatic increase in progenitors and hematopoietic stem cells driven by this metabolite 2-hydroxyglutarate. This leads to the change in hypermethylation in the DNA, and that we found that 50% of the changes in hypermethylation actually correspond to the changes found in AML patients with mutations of isocitrate dehydrogenase. Also leads to actually histone changes, and it affects the Notch pathway, the Wnt pathway, the TGFR pathway. So to end, I say that I think this is the beginning, and more genes in metabolism are actually going to be discovered. You can see 2 groups at MIT last year discovered that 3-phosphoglycerate dehydrogenase is required for many breast and colon cancer to continue to grow. Now, it is not my job to predict the future, but I think since these metabolic pathways are more limited than the oncogenes and the tumor suppressor genes, we hope that this will be part of the future together with immunotherapy and possibly targeting aneuploidy. To end, I would like to welcome you to my hometown, Toronto, and thank all the people who made this talk possible. Thank you very much.
On behalf of the American Head and Neck Society, first of all, just a wonderful talk and great description of where things are going, very exciting. But we'd like to present you with this plaque from the New York Head and Neck Society lecture. Congratulations.
Thank you.